Hi, in this video I want to give an introduction about RNA-Seq, its applications, sequencing platforms, and RNA-Seq experiment design. I'm Behruz and this is Deep Health Learning channel. Let's get started. I want to start with central dogma. Central dogma is the flow of information in a biological cell. In a replication process, the information from one DNA can copy to other DNAs. In a transcription process, DNA can be transcribed to RNA, and in translation, the information from RNA or RNA codes can be translated to proteins. We want to focus more on transcription process. Gene expression methods. We have many genes in our cells. But how we can know which genes are active in different situations and in different cells? To answer this question, we have different tools in our toolbox. We can use PCR-based techniques like real-time PCR or qPCR. It's a very functional technique with high sensitivity, which is done in real time. However, it limits us to analyzing a small number of genes at the time and requires prior knowledge of target sequences. In hybridization-based techniques, we have microarray, which can analyze thousands of genes simultaneously. And it is relatively inexpensive per gene analyzed. But it has lower sensitivity compared to qPCR and also requires prior knowledge of gene expression. In sequence-based techniques, we have SAGE, serial analysis of gene expression. It can discover novel transcripts and also provide absolute quantification, but it's labor-intensive and time-consuming, and requires a large amount of starting RNA. In RNA, we have 5' ends and 3' ends. A technique, we call it CAGE, Cap analysis of gene expression, focuses on 5' end of the transcripts. It provides information on the transcription start sites, and can be identified novel promoters in the transcripts. But it's a specialized technique and not as widely used. Another technique that we want to focus more on it is RNA sequencing or RNA-Seq or RNA-Seq. It can provide a comprehensive analysis of the transcript, detect novel transcripts, and analyze both coding and non-coding RNAs. RNA-Seq also has some limitations. It's costly, and it requires complex bioinformatics analysis. But don't worry, in the following videos, we want to learn it step by step together. RNA sequencing. RNA-Seq or RNA sequencing is a technique that uses next-generation sequencing or NGS to sequence the transcriptome in a biological sample. It provides a comprehensive view of all expressed genes and revealing which genes are active and to what extent. RNA-Seq is important because it offers a more precise measurement of transcript levels and also their isoforms compared to other techniques like microarray. Let's see what application RNA-Seq have. The first application that I want to mention is gene expression profiling. This is like taking a snapshot of all genes that are active in a cell at the given time. It helps scientists understand which genes are turned on or off in different situations, such as in a healthy tissue versus in disease tissues. Researchers can use this to see which genes are more or less active. Identification of novel transcripts and isoforms is the second application of RNA-Seq. Imagine a gene as a recipe that can be slightly altered and make different dishes. RNA-Seq can find new versions of this recipe or here transcripts that haven't been seen before. And another application of RNA-Seq is isoforms or identification of novel isoforms. But what is it? In a process that we call it alternative splicing, one RNA can change to different versions, with these different versions we call it isoforms. RNA-Seq can help us to detect these different isoforms or novel isoforms. The third application for RNA-Seq is detect of gene fusions. Sometimes part of two different genes can accidentally stuck to each other like two pages of two different books. These are gene fusions and can be important in some diseases like cancer. RNA-Seq can find these unusual combinations. Another application is analysis of allele-specific expression. Each person has two copies or alleles of genes, one from each parent. Sometimes one copy is more active than other. RNA-Seq can help identify which copy is being used more in a particular situation, which can be important for understanding some genetic disease, or also trade. And the fifth application of RNA-Seq is pathogen detection in clinical samples. RNA-Seq can be used to study viruses, bacteria, or other pathogens in a sample from a patient. It can detect known pathogens and even discover new ones. It can help doctors to understand infection better and helping them to choose the right treatments. Types of RNA-Seq assays. We have different kinds of RNA-Seq assays. Whole transcriptome analysis analyzes all RNA in a sample. mRNA sequencing focuses on protein-coding mRNAs. Small RNA sequencing targets microRNAs or other non-coding RNAs, or small non-coding RNAs. And single-cell RNA-Seq profiles transcriptomes of individual cells. Let's see which platforms do we have for sequencing. When designing an RNA-Seq experiment, selecting an appropriate sequencing platform is crucial. Here are some key points about these sequencing platforms. The first platform I want to mention is Illumina platform, which is dominant at the market for short-reading sequencing. We have short-reading sequencing platforms and long-reading sequencing platforms. Illumina is dominant in short-reading sequencing. It offers high throughput and low cost per base, and its popular models are NovaSeq, NextSeq, and MI6 series. The second platform is Torrent, which is developed by Thermo Fisher. It uses semiconductor sequencing technology, and it's based on detecting hydrogen ions releasing during DNA synthesis. It has faster run times compared to Illumina, but lower throughput, but suitable for smaller projects. The third platform is Pacific Biosciences, or PacBio. It's a platform for long-read sequencing and can sequence full-length transcripts. It's useful for detecting isoforms and gene fusions. And the fourth platform is Oxford Nanopore technologies, which are portable long sequencing read devices. They use nanopore technology to sequence single DNA or RNA molecules. It offers real-time sequencing and useful for field application and rapid sequencing. When we want to do RNA-Seq experiments, at first we have to design our experiment. To do that, we have to ask some questions. The first question we should ask is, for what application do we need RNA-Seq? For example, we want to profile gene expressions of a sample, or we want to identify novel transcripts. After that, the second question comes. The second question is, what type of RNA-Seq assay do we need? For example, we need a whole transcriptome sequencing, or just a single sequencing, or another type of assays. And the third question is there any similar experiment that we can benefit from it? And the fourth question is, how many samples and replicates should we have? To answer the fourth question, we have to consider that in the RNA-Seq experiment, the number of samples and replicates is crucial for ensuring reliable and statistically valid results. We have two kinds of replicates, biological replicates and technical replicates. Biological replicates are separate samples from different individuals or organisms at the same time. For example, if you are studying a disease, biological replicates would be samples from different patients with that disease and the samples from different persons without the disease. At least 3 biological replicates per condition are generally recommended as the minimum to perform any inferential analysis. This allows for basic statistical analysis and helps account for biological reliability. For some robust and reliable results, especially when identifying differentially expressed genes with high confidence, it's often recommended to use at least 6 biological replicates by condition. Another replicate is technical replicates. They are repeated measurements of the same biological sample. If we consider the last example, technical replicates is, for example, we take 2 samples from one person which has the disease and 2 samples from the person without the disease, or 3 samples. Technical replicates are not as critical as biological replicates, but can help assess the technical reliability of the sequencing process. However, they are not a substitute for biological replicates. RNA-Seq Workflow. The first step is RNA extraction and quality control. The second step is cDNA library preparation. The third step is sequencing, for example, using Illumina for short reading sequencing. And the fourth step is data analysis, which included itself complex computational steps, and we have to use different platforms, different analysis, different visualization tools, and also machine learning algorithms to do these steps. In the following videos, we want to focus more on this step and learn it with each other.